So in this tutorial, we'll do an assessment of the protein structure quality that we just obtained from the protein data bank via the Ramachandran plot feature in PyMol. So Ramachandran plot makes a graph of the residue backbone dihedrals, phi and psi in degrees, in terms of the secondary structure content, or let's say the secondary structure quality. So once you resolve an X-ray crystal structure, these plots help you assess the quality of your model, such that any outlier from the allowed regions will require further scrutiny, unless these regions are, let's say, occupied by residues of glycine or proline. So in case of glycine, it does not have a side chain, which allows them to attain several positions in the plot, or let's say mostly the turn or the loop regions, whereas in case of proline, they have this five-member cyclic side chain, which poses steric restrictions in their movement, mostly occupying the phi region. So there's some restrictions which allows them only to occupy a certain region, which is at a phi angle of negative 60 degrees. So before proceeding, let's go ahead and install the plugin that we need to generate the Ramachandran plot. So it's called DynoPlot, so which you can find in the plugin manager. So once you go to plugin on the menu on top in PyMol and then click on plugin manager, it will lead you to this page where it will show you the install plugins, the new install new plugins, and then settings and about. So you go to install new plugins, and in the URL, type in the address from where you're going to install the plugin, or let's say paste the address that I've provided on the slides. So once you do that, you'll have to click on fetch, and it asks you PyMol will now download executable code from the internet. Proceed. You say yes. And in the next window, it shows in which directory. So it asks you in which directory do you want to install the plugin. So the default that I have out here from my Mac system is applications. Inside the applications folder, I have this PyMol app. And then further inside that, I have the site packages for Python 3.7. So in your Windows operating systems, you should have a similar address where you can go ahead and install it. Just keep it default and let it do the thing. And so you go ahead and then press OK. The next thing that you see is plugin DynoPlot has been installed. So you press OK again. Let's close this one. And just to have another check that the plugin is really installed, you go back to the plugin, go to the plugin manager, and then go to install plugins. And in the filter field that you have here, just type in DynoPlot, that's DynoPlot, and you'll find that the DynoPlot plugin is installed and it is ready to load on startup. Okay. So the next thing that we'll do is load up a structure or the structure of the complex that we are interested in. That's the gelselin actin complex with the bound calcium ions and the water molecules and the ATP. But so there are two different ways of doing that. I have shown you the traditional way where you go to file and then click on open, and then you go to the respective directory where you have the structure. I have workspace one, and then you can open this 3ffk.pdb structure if you have there downloaded already from the protein data bank. But let me show you a fun way of doing it. Like PyMol has this interface with the protein data bank repository where they can just, you just locate a structure with their PDB ID or the PDB code, which is 3FFK in our case, and it automatically loads it directly from the site itself, from the repository itself. And all this happens on the fly. So it will not be installing it will not be downloading the PDB as a whole, but it will still show it out here. So the command for doing that from the command line interface in PyMol is fetch, fetch, and you just type in the PDB code, which is 3FFK. So do that and press enter and see the structure is loaded. So all the gelselin and the actin complex residues, that is the protein residues, are shown in the ribbon representation, whereas the calcium ions are shown in van der Waals representation, and the water molecules are shown as dots, whereas the ATP is automatically represented as licorice. So that's great. But before generating the Ramachandran plot, we'll need to display the sequence of this protein and the complexes together. So that's because it will be easier then for us to navigate via the sequence and then select certain residue positions or certain residues which we are interested in deriving from the plot. So we'll basically go to display and click on sequence, and it will show the sequence in space. So you see everything is colored in green because we didn't color them separately, the chains or anything of that sort. We'll do that in a minute when we generate the plot via representation of the chain and the specific secondary structure elements. So another fun thing or an interesting thing you can say is that since we fetched the structure directly from the protein data bank repository, so it has all the information that was stored in the PDB file. Whereas in a generic PDB file, it's difficult to retrieve the information, but this is a special kind of file that they got from the PDB, which is obviously the asymmetric unit, but it also contains elements of the biological assembly, and the extension for this one is mmcif. So if you do not provide an extension while doing a fetch command, then it will automatically take it for mmcif format, and that has this information on the missing residues, which you can see in gray out here. So the first 1, 2, 3, 4, 5, first 5 residues are missing in the gelselin protein, which is the chain A. And you can figure out if you go back to the PDB file, and you'll see that the sequence of residues of the amino acids start from residue number 28, which is exactly what is found out here with the greens. So the green ones are the ones which are reflected or represented on the screen here, whereas the gray ones are the ones which are missing. This is a very useful information. So if you go down further, you'll find that towards the end of the gelselin chain as well, you have few of the residues missing. Towards the beginning of the actin chain, that is chain V, you have few residues missing out here, which are represented in gray. And further down, you have few other missing parts. So this information is very useful because in the next tutorial, that is advanced characterization by X-ray methods, we'll be reconstructing these missing residues via an ab initio scheme by manually curating the missing regions using the build and the sculpt tool that is available in PyMol, and also by homology modeling, where we'll be using the available PDB structures which have a high sequence identity as templates to model these missing regions. So it's going to be a lot of fun. Okay, let's cut to the chase, otherwise we keep on digressing to this amazing other capabilities of PyMol. So the Ramachandran plot. So in order to generate the plot, we'll have to use the command line interface again. So if you remember from a previous tutorial, we changed a residue name of the ligand and changed its chain ID by using the alter command. Here we'll use a different command. It's called Rama, which is activated via the new plugin that we just installed, the DynoPlot plugin. So first we'll do a generic plot of all these residues that are represented in green out here. Then we go on and make a chain-wise plot. And finally, we do a secondary structure-wise plot with the specific helical regions and the beta sheet regions and the turn or the loop regions here. Okay, so in order to generate a plot on specific residue types or specific residues, we need to have a selection out here. So you could either define a selection previously, but what we'll do out here is basically plot for the whole protein together. So if you type in Rama followed by all and then press enter, it will generate a plot in a new window with all the residues that are there, all the protein residues that are here. And you have to bear in mind that the calcium ions and the water molecules and the ATPs are not represented. So this is a plot for only the amino acid residues. And each of these dots or points in the plot represent one amino acid residue and their corresponding phi and psi angles. So on the x-axis, you'll find the phi angle, whereas on the y-axis, you'll find the psi angle. As you can see, there are 4 quadrants in the plot. And if you remember from the slides, so the left top quadrant basically belongs to the beta sheets, and the bottom left quadrant basically belongs to the right-handed alpha helix, which is more common in protein residues, whereas the top right quadrant belongs to the left-handed alpha helix, which is not very common, and you can find it evidently from the less population of the amino acids out here. So let's have a look at the symbols now. So the triangles that you see out here basically represents the glycine residues. The squares that you see represents the proline residues, whereas the circles represent all the other amino acid residues apart from the glycine and the proline residues. So as you can see, evidently, the glycine residues are all outliers, or they seem to be apparently as outliers. But the main reason being that since they do not have a side chain, therefore they can occupy anywhere and everywhere on the plot, basically occurring in these regions, which are mostly belonging to the loop or the turn regions of the protein, which are highly flexible. And then you find a few outliers of the common amino acid residues apart from the glycine and proline around here, as well as you see with the round symbols here, and also a proline residue that belongs to this region. Again, here's the thing with the proline residue. Although its side chain is cyclic and it's supposed to be highly steric in nature and occupy basically a region around negative 60 degrees angle on the phi axis, which is the x-axis, which you can find out here with all the squares. Sometimes what happens is that there might be some proline residues which might occupy in these regions, which basically belongs to the coil or the turn, the more flexible regions of the protein. The reason being, it's easier for them to move around in that region, even with that sturdy or sterically hindered side chain. Now let's have a quick look at the different positions that they might occupy. So let's pick one glycine residue from here. Let's say this guy. Now, this is going to be a bit tricky out here, and you got to be very, very careful while picking this particular residue of glycine, because there's this thing with left clicking, or when you click the left mouse button on top of this, so it'll basically try to change the phi and the psi angles according to your needs. Like even if you don't drag along and just click on it by pressing the left mouse button. So never do that unless you're absolutely required to do it. So what we are going to do with this is basically go on and press the right mouse button. So that'll do the selection for us and specifically locate towards that region on this structure, on the protein structure, where this particular residue of glycine is found. So we'll just do a check here whether we are in the what selection mode are we in. So we can see that we are in the residue selection mode. So any other selection mode, like let's say atoms or objects or chains, would not work. And you also have to be in the 3-button viewing mode as opposed to the editing mode. So once you click on these, left click on these one by one, it will keep on changing the modes, but you don't want to do it out here. So okay, let's go ahead and represent this particular residue of glycine. So we'll go ahead and click the right mouse button. So once you do that, you'll see that the glycine residue of interest comes into focus. So it should be somewhere around this region because it comes into the middle of the screen in a visualization. So what you got to do is just focus on that residue, like zoom into it. So the way you can do it is, and you already know this, is basically right click, that is click the right mouse button and hold it and drag it along towards yourself so that you could zoom into it. And the residue that seems to appear at the center of the screen would be the residue of your interest. That's the glycine residue. That's somewhere around here. So don't rotate or translate the molecule. Just be there as is. And as soon as it, translates itself to bring that residue into focus, you just zoom into it. Now, let's try with the proline residue. So go back to the dynamic angle plotting, and then let's see this residue, which seems to be somewhere close to the 0 angle out here. So you go on top of this, point the mouse on top of this, and then right click on it. So you see it locates towards that residue, which should be somewhere around here, which is another loop region out here. So this is ending in a loop region. So as you can imagine, so this loop region is dangling on into the space, and it's not connected with any other region. So which means that it's more prone to fluctuations, and therefore the proline residue, rather than being in its designated location of, let's say, minus or negative 60 degrees angle on the phi axis or the x-axis, it is located somewhere around here. Let's try the same with, let's say, another amino acid residue, something else like the circles here. So if you right click on this one, it will further locate to a region. If you right click on this one, it'll further go down here, which is basically another loop region somewhere here. Okay, so this is another residue apart from the glycine or proline in another loop region. So you might ask the question, so why is it considered as a loop region when you already know that the designated beta sheet region for the, let's say, the other amino acids are basically in this region? So as you can see, this particular residue out here that we selected is slightly an outlier from the designated beta sheet region, which is highly populated around here. So the red region is around here, followed by the yellow region. So this lies somewhere around this region, slightly away from the designated beta sheet region, so occupying this loop region. Whereas If you go ahead and click a residue somewhere here, let's say this guy, then you will find that this actually occupies the beta sheet region, which is the designated beta sheet region out here. Okay, so that's that. So next what we are going to do is basically go ahead and designate it by chain or color these residues, all of these residues by chain. So I'm just going to close this for the moment, close the dynamic plotting that you had already open for the moment, and then you go back to the, so you zoom out of the structure and bring it to a position where you're comfortable with without rotating or doing anything else, because we were in a comfortable position with the structure. And you go to the selection 3FFK. So here's another thing. If you click on it, the structure will disappear. If you click on it again, the structure will reappear. So any option in this panel adjacent to this 3FFK will correspond to this whole structure out here, the PDB that we have just opened. So you have all these options A, S, H, L, C. So the C here, and as you can see from the icon itself, stands for the coloring method. So we click on this once, and then we can color by element, by chain, by secondary structure, by representation, and a lot of different things. You have different color options. So what we'll do is we'll color it by chain first. So let's go for the default option. That's the first one, by chain. It has its default coloring method. And once you press on it, we'll find that the whole structure, including the protein, is colored by chain. So different chains are given different colors, which is A and B for the gelsilin actin out here on the left. So A is for the gelsilin, which is in green, and B is for the actin. Whereas in case of the gelsilin actin in the other subunit, you have the gelsilin colored in purple, whereas the actin colored in yellow. So these are all the representations in chain coloration. And if you and you'll find the same representation in the sequence as well. So the A chain is colored in green again. And if you go down further along the sequence by using the scroller out here, so you'll find that the chain B is having a different color. And then further chain C is colored in, I'm sorry, chain D is colored in purple. And then if you go further down, you'll find that chain E is colored in yellow. Okay, so now we are gonna generate another Ramachandran plot based on these coloring schemes. So it will be easier for you to see the representations that are done based on the chain selections. So we'll type in the same command again, Rama, and then all, and press enter. And there you go. You'll find all the designated amino acid residues colored according to the chain. So these outliers out here, as you can see, for the glycine residues in the triangles colored in purple belong to this chain out here, which is of the gelsilin. And these yellow ones, which are in circles, and some of them glycines as well out here, are colored according to the actin chain out here in the other subunit. And then you have the other color representations for the different chains as well. So another thing that we can do is, and this will be very handy, is that basically coloring it according to the secondary structures. And so that it's easier for you guys to recognize based on the residue types as well as the corresponding secondary structure that it occupies. And this is kind of a cross-check with what we already did before when we didn't color it at all and had a monochrome kind of color or a single color that's green. And we basically went ahead and clicked, that is right-clicked on different residues to get their particular positions by bringing them into focus. So we'll close this for the moment and go for a different coloring method. That's, so you go to the panels on 3FFK and then click on C again, and then let's say color by SS, which is the secondary structure. So let's go for the first option because it will be easier for us to distinguish, which is helix for red, and then sheet for yellow, and loop region or turn region for green. So click on that and you find beautiful coloring of the secondary structures. So you see all the helix are colored in red, and all the sheets, the beta sheets and the beta strands are colored in yellow. And then you have the loop regions and the turn regions all colored in green. So we'll do the same again, generate another Ramachandran plot. So type in Rama all and press enter. There you go. You have all the coloring according to their secondary structures and the designated residue position. So the triangle again stands for the glycine. And as you can figure out, most of the glycines out here are loop regions because they are colored all in green. And some of the glycines belong to the helical region, These are like an exceptional case. And then you have one of the glycines that's actually beta sheet. Rest of them, most of them are all in the loop region that's colored in green. You have some of the normal amino acid residues apart from the glycine and proline colored in green as well. So that means they belong to the loop region. They can belong to the loop region, that's fine. And then you have the proline residues. In fact, all of them belonging to the loop region, which is fascinating. And then you have this guy that we showed just there while representing it, annotating it by right-clicking it. We found that it belonged to the beta sheet region. And this is kind of a confirmation that this residue indeed is belonging to the beta sheet region. All right, moving on. Another question that might come into your mind is basically how to save these figures. So PyMol allows saving figures in the PostScript format. That's with the extension PS. So you need some kind of a Adobe Acrobat Reader or something that reads a PDF file in order to open those pictures or images, or you can change it to, like you can convert it to a different format, let's say a TIFF format or a PNG format or even a JPEG format for that matter. So in order to generate a picture of this, you have, let's say, a high resolution picture. What you can do is just close this again, and then we type in Rama all followed by none, which means we are not doing any kind of selection out here, and then comma, that's followed by, let's say we give it a name, let's say Rama underscore plot dot ps. So once you do that, and if you're in the same workspace directory from where you open the PyMol along with the structure, you'll find that Rama underscore plot dot ps, that file is saved inside that directory, and you can open it with a PDF viewer, and then you can convert that into different formats.